our Module 3 project. We are Group B14, and our topic was activation of brown fat in adult humans. Brown adipose tissue, or brown fat, has a rich supply of blood as well as nerves, unlike white fat. The percentage of brown fat in the body decreases with age. Brown fat produces heat and allows mammals to live in sub-thermoneutral conditions without having to rely on shivering. Brown fat is healthier than white fat because it produces lots of heat by burning calories. White adipose tissue, on the other hand, acts as an energy repository and is unhealthier than brown fat. Thermogenesis is the process of heat production in organisms. Non-shivering thermogenesis occurs in brown adipose tissue. There are two types of thermogenesis. The first is obligatory thermogenesis, which is when heat production automatically occurs because of metabolism. The second type is facultative thermogenesis, and that can be activated with cold exposure. The challenge is to figure out how we can turn on brown fat, or even turn white fat into acting like brown fat to fight the obesity epidemic, as well as just make us healthier.
There are two scientific tests to approach the conclusion. The first is Journal of Clinical Investigation. Scientists look beyond brown fat's heating-producing capacity or how, once activated, it affects our metabolism. With a sample size of 6 healthy men, they reduce average skin temperature by about 4 degrees to roughly 30 Celsius by fitting them in a cooling suit. This allows scientists to see, for the first time, that cold exposure increased the amount of non-acidified fat acid in the bloodstream by 1/3. The second test is PNAS, stands for Proceedings of National Academy of Sciences. In this study, researchers tried three masses on separated ways once a course of 3 days. 10 participants received a single dose of ephedrine or saline solution, or were fitted with a cooling vest set to 40 Celsius. Such vests are sometimes worn by surgeons in the operating room. They found that brown fat was reliably activated once during cold exposure, not by hand.
In a second study focusing on the metabolic abilities of BAT, researchers used PET scans to monitor oxidative metabolism, glucose uptake, and fatty acid uptake using chemical indicators. These three measurements are representative of the metabolic activities of BAT. Each of these measurements were taken at several different muscle groups, as well as in BAT. BAT cells were traced by a protein known as UCP1, which is known to be responsible for much of the function of BAT. Here we have the results of the experiment when testing the glucose uptake of BAT and the surrounding muscle groups when cold conditions are applied. As you can see, the glucose uptake of BAT significantly increased compared to almost all of the surrounding muscles, with the exception of the longus colli, which is a muscle on the back of the neck. The same is true for the NEFA, or non-esterified fatty acid, uptake in BAT. The fatty acid uptake in BAT significantly increased compared to the surrounding muscles under cold conditions. This plot shows the change in the rate of oxidative metabolism in different muscle groups under normal, represented by the red line, and cold, represented by the blue line, conditions. Note that in the central top graph, the rate of oxidative metabolism of BAT is significantly higher under cold conditions, whereas most other muscle groups show no such increase. To understand exactly how thermogenesis works and why it can help lead to weight loss, consider the process of oxidative phosphorylation, in which energy from the electron transport chain is used to pump protons across the inner membrane of the mitochondria to create a proton gradient. In oxidative phosphorylation, the protons then flow through the enzyme ATP synthase, represented in the picture by the pink structure, to produce ATP. However, in the process of thermogenesis, protons travel across the inner membrane without passing through ATP synthase. Thus, less ATP is produced, and the energy that would have been stored as excess weight is lost as heat. There was an increase in the activity of oxidative metabolism in the BAT, but not in surrounding muscles. There was also an increase in the fatty acid uptake in BAT, which presents a potential opportunity for weight loss. Energy expenditure was measured to increase by 80% over the period of cold exposure. Because the oxidative metabolic activity of BAT increased relative to the other muscles, it is likely that BAT activation led to a large fraction of this energy increase. This means that BAT plays a vital role in thermogenesis. In conclusion, it may be possible to activate brown fat in adults through exposure to cold conditions. This idea is supported by both studies. The first study indicates that the drug treatment has not succeeded in activating BAT, although there is room for future research. The relevant implications of activating brown fat in adults are that the oxidative metabolism will increase in speed and that the fatty acid uptake will increase, both of which may be able to contribute to weight loss.
So the take-home message is that brown adipose tissue is more effective at cold exposures. So in order for weight loss to occur, cold exposure is one of